So go ahead and start. Next slide. So talking about biodiversity in general, and I'll cover 4 topics. How do we measure biodiversity? Uh, is biodiversity really just a sample process? So there is that stochastic theme. Some work on global change, some of which is older, so I'll go through that pretty quickly. And then some newer stuff about some implications back into basic understanding of biodiversity as a consequence. So next slide. That's it. Next slide.
Again. Okay.
There we go. Yep. Yeah, so biodiversity originated, a lot of people attribute it to E.O. Wilson's book, but it had been in use prior to that. It very much came out of a policy context. E.O. Wilson came up with it for a National Academy symposium that was trying to influence policy. It definitely did not originate in a scientific context. So next slide, please. So that presents a challenge. Science is based on operationalization, the notion that things are repeatably measurable, right? If I measure something and Jeff measures something, when we come up with completely different answers, it's pretty hard to do science on that. So the repeatable measuring of things is important. So the challenge is to apply that to biodiversity. Next slide. So I'm going to just use an example here, some warblers that live outside of Orono, Maine, where I live when I'm not on sabbatical. And we see here, we've got, what is that, 17 different species of warblers. They're pretty colorful little birds. And so I want to see if we can figure out what the biodiversity of this group is. So next slide. So of course they all have taxonomic names. That's often a basis for diversity, is the species concept. Go ahead. This is just to point out, black and white is never as exciting. I think some of biodiversity is just that emotional impact. Go ahead. This is starting to get into the quantitative aspects. So it depends on which year you're looking at. If we start in 2006, 2 of those species have dropped out. If we go ahead. 2012, we have a different set of species there. Go ahead. And, if we summarize across a 10-year survey, some species are there all 10 years. One species there is only a single year, and one species, there's different flickering ins and outs of species. Go ahead. And of course, this is just looking at presence absence, but some species are very abundant. You count many individuals in a single route, and other species are quite rare. So go ahead. And this is, a full presentation of the data that's behind what I just showed you. Each of those lines is a species. Some of them are flickering in and out. Some of them are quite high abundance on a regular basis, but all of them are changing through time. Go ahead. So now it's not quite so clear if we want to talk about the species richness of this group, what's the right number? If we look at the cumulative richness, we end up at 17, which is where I started. If we look at the average richness, it's more somewhere around 11, 11 and a half. And of course, if we start to take something like abundance into account, we have Shannon diversity, which is considerably lower because most of those species are very weakly represented in the overall assemblage. Go ahead. This is plants. So these are trees in Barrow, Colorado Island. This is just 4 species. There's 225 species in this plot, but I'm just showing 4. And I just want to show how it's not just in time, but in space. Go ahead. So different transects are going to come up with very different answers about the number of species. And that's partly because these individuals are just not distributed evenly or randomly across space. Go ahead. So to summarize, well, not to summarize, but also just to acknowledge, I'm just looking at taxonomic diversities. There's all kinds of other diversity that are important too. So that just makes the problem harder. Go ahead. And the key point here is that physicists like to talk about extensive and intensive quantities. Extensive quantities, such as the number of gas molecules. Just if you double the volume you sample, you typically double the number of molecules. And you can then work with densities, number of molecules per liter per square meter, whatever you want to work with. And you get this linear scaling, and the slope of that line is the density. And you don't really have to worry too much about the area that you're looking at. Other quantities, such as temperature, are intensive. And they just average as you increase your scale, you just average across the volumes. The problem is biodiversity is neither a line, or it has neither a slope nor an intercept. It's a nonlinear function. It increases, but in a decelerating fashion with scale. So go ahead. So figuring out how biodiversity scales is going to be the key. And just to put this in context, I want to ask a simple question. Does an invasive, and this is data from John Chase and Tiffany Knight, if an invasive species comes in, does it increase or decrease the local biodiversity? Argued we have to pay attention to scale. So should we scale by sample size or number of individuals in the area? Go ahead. And the answer completely depends on if we scale by sample size or if we scale by area. If you scale by area, uninvaded plots have a higher diversity at both the alpha and gamma scale. But if you scale by sample size, number of individuals, invaded plots actually have a higher diversity than uninvaded plots at both scales. Go ahead, please. And it turns out the main effect of invasion is actually to decrease the abundance. And so that explains that result. So we need to find some way of dealing with the scaling, and whether we scale by area or scale by abundance. Go ahead, please. And I would like to argue that the solution is rarefaction curves. So these are, you've probably seen rarefaction curves before. In fact, they're a very old technology. I think it's 1967 Saunders. And they keep kicking around and disappearing and kicking around and disappearing. I'm going to argue that we should keep them around. So the two data sets that the invaded and uninvaded are nicely captured by these rarefaction curves. So the x-axis, horizontal axis, is number of individuals. As sample size increases, the vertical axis is richness, and the lines are averages across many samples. Go ahead, please. So these rarefaction curves pack a lot of information. The height of the curve at the tip gives you the raw species richness. The horizontal position of the ends of the curves gives you the total abundance in your samples. Go ahead. This allows you to do the original use of rarefaction, which I'm going to argue is much too simplistic, to just rarefy and chop things back and compare. And there you can see that once you control for a number of individuals, richness is higher in the uninvaded solid line plots. Go ahead. But there's actually a lot more information in these plots. It's been shown that the slope of the line from 1 to 2 individuals is actually an evenness metric, pi evenness, which is a hill number 2. The other end of the axis over on the right side has a hill number of 0, for those of you who know hill numbers, which kind of implies, at least in a hand wavy way, that this line also somewhere in between has a hill number of 1, or Shannon evenness captured in this curve too. Go ahead, please. And so this is showing that the top line is the rarefaction curve of a very simple community with abundances of 1, 1, 2, 5, 10, and 50, so 7 species. The black line is the slope of that rarefaction curve. The middle panel is the contribution of each species to that rarefaction curve, and the bottom panel is the contribution rescaled to 100% of contribution. And what you can see is that on the left-hand side, it's all about the very common species, and on the right-hand side, it's all about the very rare species. So you actually, going with the theme of rarefaction curves, packing a lot of information in, you can actually, if you get good at looking at them, you can tell a lot about what's the structure of a community looks like. That one curve contains not just total richness, total abundance, evenness on the left-hand side, Shannon evenness somewhere in the middle, but you can see the proportions of rare and common species as well. Go ahead. So the number of species that actually show up in a sample is a function of three things: the size of the sample, the spatial structuring or clumping of individuals within a sample, and the relative proportions of rare and common species. Go ahead. And that's very clear in the BCI tree data. The role of the clumping makes it very clear that if they're more evenly spread, you're going to encounter more species quickly. If they're clumped, you're going to encounter species more slowly. Go ahead. And here's Jeff's stochastic. I'm going to use the metaphor of not gases in a volume, but marbles in a jar, with different colors being different species. Go ahead. So working with John Chase and Nick Catelli and a bunch of people, we've produced an R package that takes rarefaction curves and has developed a set of null models that lets you tease apart the contribution from each piece, the contribution from density or abundance changes between. So this is designed to compare a treatment and response of either experimental or observational data. So you're comparing two situations or treatments, and it can tease out the contribution of abundance, the contribution of spatial clumping of individuals, and the contribution of the proportion of evenness or proportions of rare and common species. You see that in the three different curves, and then you still have the rarefaction curves, so you can actually see scale changes. So at small scales, the aggregation effect is very important, but as you go to larger scales, that effect disappears, and actually just the simple abundance, the blue line, becomes the most important effect. And it's a negative effect, whereas the change in common versus rare species is actually a positive effect. So those are all counterbalancing each other out. So this package is an R package. It's called MOBR, Measurement of Biodiversity in R. Go ahead. Okay, so hopefully I sold you on rarefaction curves. The thing I want to caution you about rarefaction curves is we're very used to simple 1 or 2 parameter curves like the log curve, the power law curve. Rarefaction curves actually have as many parameters as they have number of species, and they're extremely flexible. This shows the extremes, right, where we have one individual of each species, and the curve can only go up because we're always encountering a new species. At the other extreme, that dotted line at the bottom right, they're completely clumped. So we go all the way through all the individuals of one species before we get a new species, and we keep going. Of course, the real situation is somewhere in between, but these curves are extremely plastic and bendable. Go ahead. And what the consequence of that is these curves can cross each other. This is a sample data set of marine invertebrates, and of the 120 pairs of curves that we have there, 53 of those 120 curves actually cross each other. And if you go over to the right-hand side, 53 cross at least once. It looks like about 8 cross each other twice, and we actually have 3 or 4 curves that cross each other 3 times. And what's it mean when those curves cross? That means there's a scale dependence to the answer, right? When the rarefaction curves cross, it means at one scale, one curve is higher and has a higher species richness, and then after they cross, it flips and the other curve is higher and has a higher species richness. So not just the magnitude, but the sign of changes in richness can actually completely change depending on scale. Go ahead. This is a meta-analysis of a bunch of studies of various types of global change impacts. And so we have there's in each case, there's a plot scale, and then there's the aggregate scale of all the plots put together. And this is just looking at the effect, log effect ratios on species richness on the small scale on the horizontal axis, large scale at the vertical axis. Go ahead. And 2 things to note. One is that the line that fits is got a slope of less than 1 to 1, which means that on average, the effect size is weakening at large scales, the richness effect. That's maybe not so surprising, but those red boxes contain studies where the effect of, let's say, a disturbance or an invasive species or fragmentation completely changed sign, and it's positive at the small scale, and it has a negative effect at species richness at the large scale, or vice versa. So scale dependence is critically important when you're talking about species richness. And again, I would argue rarefaction curves are a great way, because scale is specifically built into the x-axis, a great way to acknowledge and address the scale dependence. Go ahead, please. And unless you think this is all kind of theoretical, this is a nice paper by Jack Lennon and others plotting species richness of birds from British bird surveys and scales. So on the left side is the original raw data, 10 by 10 kilometer pixels. And on the right-hand side, we have 90 kilometer pixels. I don't know if you can see, I can't mouse over it, but you might know where London is. It's that ellipse that's down at the bottom, just to the left of the notch of the Thames River. And you can see that actually at the small scales looks quite diverse, especially its southern edges. It's got some of the highest richness. And if you look at the black down below, those dots are the highest, I think, 5% of richness in the entire island. and some of those occur in London. But when you go over to the right-hand side, species richness has completely collapsed, and it's basically all in the Lakes District National Parks area up in northwest England. And so the very answer about what is a diverse or a non-diverse place actually depends on the scale at which you measure it. Go ahead. Okay, to summarize, biodiversity changes can actually flip sign depending on scale. You have to be scale explicit, and rarefaction curves are a great way to do that. And we have developed methods that let you actually tease apart some of the contributors to richness, including the proportion of rare species, changes in abundance, and changes in regional richness. Go ahead. And also spatial aggregation. All right, now we're going to go further down the stochastic road. Go ahead, please. And typically, one more advance, please. Yep, typically this is how ecologists like to think about things. We're very reductionist. We like to work from small pieces going up. And so we think of processes at the local scale explaining regional, which explains biogeographic, but go ahead. I'm gonna just take it as a thought experiment here and go the opposite direction. What if we think about all of the interesting and deterministic and explainable action happening at the large scales and the local scales just being samples? Go ahead. So, you know, humor me in a wild thought experiment. We're going to go back to the colored marbles. Each color is a different species sitting in a jar, and we have a regional species pool with different abundances, and the local community is just a sample from that regional community. And of course, different local communities might have different sample sizes with more or less individuals. And the species is really just a consequence of how many different colors of marbles that sample pulls out of the large jar. Now that seems like a wild, crazy idea that can't possibly be true. It's kind of pretty explicitly ignoring species interactions. It's probably ignoring environmental fit and a bunch of other things as well. But let's give me some rope and let's see how all this works. Go ahead, please. So I'm not going to go through all these equations, but, and this is work that's been kind of building over time through a lot of different papers, but there's one core function known as a sampling function. It's up there at the top center, the sigma function, which is the probability of sampling. Yep, you're dancing around it there, Jeff, thanks. Sampling little n individuals from a regional pool that has big N individuals, and the big A and little a are the area of the local sample in the regional pool, which is really just to help us get a scaling between the two. And then the question is, how many parameters do we have to add to that function to describe the ecology of a local community? That's theta being the parameters. So that's the question I want to pose. Over on the right-hand side is a graph of what that function looks like in the BCI tree data. So across the horizontal axis is bins of abundance, and the vertical axis is the number of sites where each of those graphs is a different species. The number of sites where that species has an abundance of 0, 5 to 10, 10 to 15, 15 to 20, etc. A null model, a complete randomness, would be a Poisson distribution, which would be the black line. So let's see how well that works. And the point is, once you have the sampling function, the species area relationship, the local species abundance distribution, the abundance occupancy relationships, local occupancy, and even beta diversity are all just direct functions of the sampling function. So all of these patterns that have kind of long been considered really important in macroecology and community ecology boil down to that sampling function. So let's see if we can, again, the null model of marbles in a jar would be that the sampling function is Poisson, and we don't have, so the only parameter is the abundance in the regional pool, and we don't have any parameters. So let's see how all that works. Go ahead, Jeff. It works pretty well. The top left panel there is a species area relationship, number of species versus area. This is BCI data. The black line is the observed, and the blue line is what's predicted by this model with Poisson random sampling. The top right is just showing the error in number of species. The bottom left is a histogram of the species richness, the alpha diversity in each 1 hectare plot. The true answer average is 91. The predicted average using this binomial Poisson sampling is 103. In ecology, is predicting a richness of 103 when the answer is 91, is that good or bad? I don't know. The way ecological models go, I tend to think that's pretty good, but it's clearly not perfect. And there's the prediction of occupancy versus the actual occupancy of each species. You can see there's some systematic error here, right? We're regularly over predicting richness and we're over predicting occupancy. And so there's probably a systematic explanation for this. Go ahead, please. And the answer is that individuals within a species are clumped. So those 4 left graphs are 4 different species of trees in BCI. They all show a form of clumping, meaning individuals within a species are closer to each other than random. Also, clearly they're not uniform, right, evenly spread in a grid, but they're also not random. They're gathered into clumps. And we could see that really already in those graphs I showed you. Those graphs have an excess of zeros and an excess of very high abundance. They're over dispersed relative to the Poisson distribution, and that's a function of the clumping. So replacing the Poisson, there's a distribution called the finite negative binomial distribution by Tommaso Zilio and Fenglang He, which was published. And so let's replace the sampling function with the finite negative binomial. It adds 1 parameter, which is the degree of clumping. So go ahead, Jeff. This is a ridiculously over-parameterized model. It has a clumping parameter for each species and a different clumping parameter for different scales. So it works well, but given the number of parameters, maybe we shouldn't be too surprised. But you can see the species area relationship is close. Our alpha diversity is now much closer. The problem was we were assuming the individuals are spread out evenly across those 50 hectares, but they were actually clumped into a few of the hectares. And so that's why we were and most of the time we were missing them. And they were only showing up in a few hectares. So that's why we were over predicting richness. Now we're quite close. And the occupancy is, perfect prediction, or R squared 0.99. I don't remember exactly, but it's a very strong prediction. This is badly over parameterized. Let's see if we can do a little better. Go ahead. So this is just 2 parameters. This assumes that 1 clumping parameter across all species. So no different clumping for species, kind of an average clumping for species. And assuming K varies according to scale. So this is just fitting a line, a linear regression of K versus the average clumping for a species. And you can see this does almost as well. The species area relationship is extremely well predicted. The local alpha diversity is extremely well predicted. And the occupancy is not quite as well predicted, but we no longer are systematically over predicting. We're coming pretty close to predicting occupancy. Go ahead, please. So hold on this graph for a sec, but if you just go back to that page, I had the sampling function, and I said, how many parameters do we need to make this work? The short answer is no parameters. Complete randomness works pretty well, at least as ecological models go. And adding 2 parameters that describe clumping with scale explains local community structure, species richness, occupancy, beta diversity, etc., extremely well with just those 2 parameters, a generic clumping parameter and how that changes with scale. So go ahead, Jeff. So on the left side, up to scales of about 100 by 100 kilometers, these random marble models work quite well and predict species area. The background figure is the triphasic species area relationship from Preston, who Jeff already mentioned. And up to scales about 100 by 100 kilometers, it predicts this increasing decelerating species area relationship, which matches the left-hand side of the triphasic relationship, and it's really just marble sampling. The right-hand side, go ahead, has a very similar model behind it, where we're now, instead of dealing with individuals, we're dealing with species ranges, and we're randomly placing down species ranges of varying sizes and heights, and that predicts the right-hand side quite well. So go ahead. So to summarize that, the local scale arguably is just a very noisy echo of the regional scale. And that suggests we should be studying biodiversity at larger scales, the mesoscale, the kilometer, the 100-kilometer scale. And when we do that, I think that's good news. Some processes are changing. The amount of stochasticity decreases, right? N is going up. Environmental determinism becomes more and more relevant. microclimates and behavioral responses and things start to disappear. Species interactions don't disappear, but they become more diffuse. I don't compete with this one exact species. I compete with a guild, right? And in one location, I may be competing with species A, and another location with species B. But more broadly, I'm competing with the guild of warblers or woodpeckers at larger scales. At these regional scales, evolution and speciation start to become important, and dispersal becomes a little more manageable. We are no longer dealing with mass effects and source sink dynamics, and really just dealing, Jeff mentioned island biogeography, we're just dealing with colonization, extinction dynamics at larger scales. But I also argue working at larger scales, as implied by this kind of sampling paradigm, is convenient because that's really the scale that conservation works at and should be working at. It's the scale of policy, for sure, but it's also the scale at which we conserve and manage species. So go ahead. I'm going to switch gears for a minute here, and then I'm going to try and bring them back together. So we've been conducting a giant experiment, really, for at least several 100,000 years, but especially the last couple hundred years, and especially the last 100 or 150 years, where humans are impacting the planet. What's the effect this is having on biodiversity? Go ahead. So I follow the Millennium Ecosystem Assessment Framework, 5 major types of global change. Land use change on the left, harvesting and potential overharvesting in the top center there, climate change on the bottom center. Over on the right, we have kudzu, a notorious invasive species. And on the bottom right, we have chemical change. Most people think about pesticides, which certainly are important, but PFAS is a huge thing in the United States, forever chemicals. But really the single biggest change is fertilization and eutrophication, nitrogen and phosphorus application. So 5 major global changes, go ahead. These are not small. Humans have occupied about 50% of the land. We've removed about 50% of the terrestrial biomass. We've removed about 50% of the fresh water. We have doubled the amount of nitrogen coming into the biosphere every year. We will very soon have doubled the amount of CO2 in the atmosphere. We have removed well more than half of all the fish, large marine fish, tuna and cod, things like that. In the marine ecosystem is a little more complicated. We've taken about 8% of NPPP and about 25% in coastal waters, and it's a little mixed. It's harder to say 50% of the ocean bottom has been affected by humans. It's a much more stirred pot, but kind of this amazing rule by 2. Humans have basically doubled and halved the planetary cycles. Go ahead, please. So working with Anne McGill, Maria Dornalis, Nick Catelli, we kind of posed that there's not one biodiversity trend, right? That's implicit in the E.O. Wilson use of biodiversity as a monolithic concept, and I hope I've convinced you to unpack it. This is another argument to unpack it. We argue, as I already said, scale is very important. The trend at a global scale does not have to equal the trend at the local scale. And also there are multiple patterns. There's temporal beta diversity, turnover, there's alpha diversity, there's spatial beta diversity, and there's just abundance or biomass. And this was our best guess at the trends at the time we wrote that paper, and we've actually, along with other people, been working really hard to fill out that grid in the time since then. Go ahead. First, just to acknowledge, about 50% of all trees are gone. So, numerically things are hugely impacted by humans. Go ahead. But in the areas that aren't parking lots and cornfields, that's an important condition, because this graph would look different if we were averaging across the 50% that look like that. The left graph is species richness versus time, a big assemblage of different data sets. And you can see richness is going up in some places. These are all local communities, local meaning anything from a square meter to probably 100 kilometers, but not global. And various diverse taxa, from plankton to birds to trees to mammals. The average trend is actually surprisingly close to 0. On the other hand, the right-hand graph shows the species composition, Jacquard index, basically percent of species that are the same as you move through time from the start of the sampling process to the end. And we found very fast turnover. On average, 10% of the species have changed every decade. Obviously, it's faster in plankton and slower in trees, but on average, that's a very high rate of change. So more than 50% of the species will have changed in a community in 50 years. Is best, we don't know what that looked like before humans, but against some null models, that looks like a very fast accelerated rate of change. Go ahead. Um, this is just again suggesting that the change in species richness may depend on scale. And in particular, regional scales, it may actually be going up, and at local scales, it may not be changing, and at global scales, it may be going down. I'd say might be the best current weight of evidence. Go ahead. This is a graph not at the community level, but the population level, single species at a single site. And this is the trend in population size abundance through time. Take a look at that graph. It's a nice peak graph. Looks like a normal distribution centered on 0. Go ahead. Just to bring it back to the warblers, we get the same kind of story. Some are going up, some are going down. Net trend is not a whole lot of change. Go ahead. So this is a review paper that that group and a bunch of other people put out recently. This is a pretty complex figure, but I'm going to walk through it in a little bit of detail. So the top left, we're looking at species richness, one of the 4 metrics, and we have 3 scales: global, regional, local. And if you're in the top row, you're having large increases, and if you're in the bottom row, you're having large decreases. So globally, species richness is going down. Regionally, species richness is clearly going up by a little or a lot. And locally, it honestly looks like species richness is somewhere between neutral, that middle bar, and increasing, the top 2 bars. All right, go ahead. Different metric. Temporal turnover seems to be very high everywhere at all scales, local, regional, and global. So the churn or change or turnover rates are very high. Again, it's hard to figure out what that looked like before humans. We don't have the quality data sets to do that, but strong suggestion that one of the biggest impacts humans have had is increasing turnover rates. Go ahead. Spatial beta diversity. If you take the current weight of evidence, there are a few studies that say it's neutral, i.e. neither increasing nor decreasing. But the current weight of evidence across published studies is that spatial beta diversity is going down or homogenizing. And go ahead. And abundance and biomass, we seem to be somewhere at a local scale, seem to be on average, not changing. At regional scales, now we're picking up those cornfields and parking lots, and that clearly is impacting the total abundance or biomass at the regional scale. And the global scale, it seems to be going down. Go ahead. I would suggest that there's perhaps some publication bias in the spatial beta diversity scale. This is a recent paper published. Actually, Sean Blows in the office next door to me is the lead author on this. Large meta-analysis, hundreds of data sets looking at homogenization, decrease in spatial beta diversity versus heterogenization, which is an increase in spatial beta diversity. You can see that we found the average is a decrease in spatial beta diversity, but if you look at that top right figure, it's a very, very small decline. And if you look at the bottom figures, you can see that the real pattern is that some things are declining, increasing homogenization, and other places the beta diversity is increasing, which is heterogenization. And the figure on the left, I'm not going to spend too much time on, but it's just showing, goes back to Whitaker's beta diversity and shows that there's a link between local and regional. And the figure on the right shows that most of the beta diversity changes are due to local changes in species richness, which we already know was kind of mixed, increases and decreases. So that's not surprising that beta diversity is having the same outcome. Go ahead. One more paper looking at change, looking at community average body size, community weighted mean body size of an individual. So this is not biomass of the total community, but the average biomass of an individual in a community. Change can happen 2 ways. You can have individuals get smaller without changing species composition, or you can have species composition shift towards smaller species, which is what happened in the bottom figure. So we did a big meta-analysis across many data sets. And next slide, next arrow. The bottom line is, that's all right.
Sure.
Bottom line is fish show a significant decrease in body size. Everything else is very mixed. So same kind of story. You can find increases or decreases, but if you look at plants across many data sets, some data sets show an increase in body size, some data sets show a decrease in body size. If you look at mammals, increase, decrease. If you look at fish, they're shrinking. Go ahead. And perhaps it's surprising or perhaps not surprising, but the change is more due to species in all of these, not just the average of fish declining, but all of the data sets showing change. We came up with a partition using the price equation, and you can show the change is much more due to species composition. Not much more, it's due to both, but it's clearly more due to change in species composition than body size. Go ahead. Weirdly though, even though we're seeing, this is kind of a theme that's emerging, seeing all of these changes in underlying composition, the total community biomass across these data sets is staying constant. So we're getting this churn and then this constancy, which is kind of a theme that's emerging. So I want to address that more directly. Go ahead. One last slide on global change. This is, you may have heard about the Living Planet Index, which shows a very different story from the story I showed of, this strong peak distribution centered around zero. What we found is we had 5 different models of, significant change of the entire group. The next line down is the main group is not changing, but there's a small group of extreme declines, which gives the same average. Yep, thank you. There's the arrows. And then what we found is actually that second line. Of course, you could have increase happening too. Go ahead, back. We found that second line, that the core group of species is not changing at a pretty small fraction. A couple % of species are showing massive declines, and that's driving the whole overall trend. But not that you would take those species out in a policy or conservation context, but you take them out, the remaining group is strongly centered on zero. Okay, go ahead. So kind of net, we seem to have this pattern of, increases, decreases that kind of balance each other out and show this kind of almost like a central limit theorem of human impacts. And the main effect, go ahead and hit down once, is churn. Well, first off, the main big effect is we've taken up half the land and half the trees, right? So we've taken away a huge amount of habitat. But in the remaining habitat, we kind of get this central limit theorem kind of response. Some places are increasing, some places are decreasing, but the turnover is very marked, right? So a lot of change, but not so much change. In fact, close to no change in these big picture metrics like richness and diversity. The marine system, really the main signal is just the churn. Okay, go ahead. So that's been pretty much my career for the last 15 years, is how to measure biodiversity and looking at how biodiversity is changing under human impacts. And there's some pretty surprising patterns in how biodiversity is and isn't changing. So I've been increasingly turning, and this is work with Nick Catelli and Anne McGill and Maria Dornalis. Let's step back at, how biodiversity is and isn't changing. And what's that tell us about core processes in ecology? Go ahead, please. So why do these things keep having trends that are zero, right? Central limit theorem can explain how you have this symmetric distribution, but the central limit theorem doesn't tell us that it should be centered on zero. So we came up with we took the same time series. This all comes out of a database called BioTime with over 100 different time series in them, community times series. And there's a test called the ADF, Augmented Dickey-Fuller test, for whether something is a random walk or it has a central return tendency. They can be very hard to tell apart because if you look at multiple random walks, they're going to average to a central tendency of zero. But that's different than saying within any one random walk, if it gets too low, it tends to come back. And if it gets too high, it tends to come back down. So the ADF test is a test for random walks versus actually a central tendency. And we did this on species richness and total community abundance. And anything less than zero has a tendency for regulation or return. Anything above zero is a random walk, which if you look at many ensembles is going to average to zero, but individual time series don't have a return tendency. And you can see there's a very, very strong propensity for regulation or return back towards the 0 point. The ones that are in dark red are statistically significant. The pink ones are trending but not statistically significant. So both total richness and total abundance in these time series show not random walks, which is maybe an argument against the central limit theorem, but a pretty strong return tendency. Go ahead. Not going to go through this whole graph, but we spent a lot of time trying to determine alternative explanations for these trend of 0. It's not a random walk. It's not due to observation error or detection probabilities. It's not a portfolio effect where species richness, where when one species goes right, the ADF test only works for a single time series, but we of course have many species in a community. There doesn't seem to be the strong compensation effect where when one species goes up, another one has to go down. We did not find a strong environmental tracking signal where everything would go up together and everything would go down together. We did find that the underlying environment also shows a strong central tendency or return tendency, and species do track it, but that's not what's driving the regulatory response we found. So we're really left with some core mechanisms about niche shifts and the total amount of energy available to a community and partitioning of that energy. And the niches may shift and the species may get different shares of that energy, but the energy is available to a community is what's driving total richness and total abundance. Was really where we ended up. Go ahead. And I just want to give you some examples on that. So on the left is a nice paper by Morgan Ernest et al. At the Portal small mammal site in Arizona. And they found, in their case, they did find a strong downward trend in average mass of a species. They found a compensatory strong upward trend in the total number of individuals. And the bottom left figure was the total energy flux in the community, which is constant. The figure kind of below and to the right of that cute little rodent is grassland species versus shrubland species. They had a complete turnover in the basic biome of the rodent community, going from a strong grassland community to a strong shrubland community. Yeah, thanks, Jeff. And also species richness, that graph down at the bottom, was constant. They did the same group did the study in Barrow, Colorado Island, trees. They found somewhat the opposite story. Abundance went down in that case because the tree size was going up. A lot of people don't know, but BCI is actually still a secondary growth forest. It's getting to be pretty late stage, but it was farmlands back in the 1930s. And so it's still showing tree size increase. And the total energy flux, the second graph down, is constant. Somewhat similar, this is work by my student Julie Messier, also in Panama, of functional traits of a species. She looked at leaf mass per unit area, so basically leaf thickness. And the average community value did change along an environmental gradient. The drier places had higher LMA than the wet places. But within a community, so plots that are, say, 100 meters apart from each other, the distribution of traits, so those are the thin, for example, the thin green lines, the thick green line is the average across 8 different plots, and the thin green lines, there's a highly conserved trait distribution. Even though this is the tropics, the species distribution is highly not conserved. There's only about 10% species that are shared between 2 of those green lines, but the trait distribution is highly conserved. And then down at the bottom, returning to the Martins et al. Paper, which also was done here at IDIV, you can see the fish that are significant are off there to the left. The rest of the graph is pretty strongly centered on no change. In body size, there's a compensating change in abundance. The change in total biomass is strongly centered on 0. So there's this weird kind of dynamic that things are more regulated than we might think. Go ahead. So, I increasingly have been moving in this direction, but species obviously matter for reproduction. It's pretty hard to reproduce without another member of your species unless you're clonal or asexual. But the community assembly really pays no attention to species, pays attention to species. Community assembly is completely at the individual level. It's about how many individuals can make it in, and the answer about how many individuals can make it in is a function of energy and also a function of traits. There's a optimal trait distribution for an environment, and there's an amount of energy available. But the individuals are sampled in and almost without regard to which species are coming in, which also helps to explain the very high turnover in species richness, species composition, the set of species there, even as richness is holding even. Okay, go ahead. The other thing I kind of intentionally, maybe too subtle, but asked you to look at that graph of change and kind of implied it was normal. This graph is 9 different trends in biodiversity metrics. So we have species richness in the biotime data. We have Shannon evenness, Shannon diversity, Simpson diversity. We have total abundance across the community. We have Jacquard similarity. We have we have population level trends, so single species. On the bottom left, we have a data set from Joel Kingsolver, which is selection coefficients. So this is rate of change of a phenotype. Bottom center, we have just the breeding bird survey, the population growth rates, and we have species richness of the breeding bird survey, different routes at the bottom right. And all of these graphs kind of look, well, first of all, they're almost all strongly centered on 0, which is surprising in what I was just talking about. But they have another feature. They're not actually normal. The red line is normal. These have much more leptokurtosis. They're much stronger, sharper peak at that 0. And it's hard to see. You'll see it in the next graph. But they also have much more very wide tails. The tails are, in a formal sense, fat. So you have many outliers and a very sharp peak, and very few things that are just, well, compared to a normal, fewer things that are just a little bit high or a little bit low. You're either going to stay right close to 0 or you're going to go crazy and go way out to the ends. Okay, go ahead. Turned out a Russian probability theorist anticipated this back in the 1920s by the name of Subotin. He tried to think about errors and error measurement, and kind of basically, in a very polite way, argued Gauss had got it wrong, and it doesn't have to be normal. It has to fit a Subotin distribution, which is of the form e to the minus x minus mu to the alpha. And of course, in the normal distribution, alpha is 2. But Subotin argued that actually error distributions can be much more general, where alpha can vary from 0 to infinity. And on the left-hand side, you can see the normal distribution is when alpha equals 2, the black line. But there's also something I want you to home in on called the Laplace distribution, which is the red dotted line, which is alpha equals 1. So there's an absolute value in that alpha, that raising things to the power of 1, which makes it symmetric about mu. But there's a whole range from completely platykurtic to completely leptokurtic distributions. And Subotin said, from a theoretical point of view, there's no reason to prefer any value of alpha. It's an empirical question what the value of alpha should be. So go ahead. I'm sorry, Jeff, go back one. I wanted to point out on the right-hand side, we used a log frequency scale on the vertical axis. And so the normal distribution becomes a parabola, makes sense, x to the x squared. And the Laplace distribution becomes the red dash becomes a triangle, a tent-shaped function, which makes sense because it's a plot of the absolute value function, basically. Okay, so go ahead. So here's my 9 data sets versus the Laplace distribution. The error bars are Monte Carlo simulation of error bars. And you can see every one of those data sets conforms extremely well to the Laplace distribution. Even out in the tails, which is pretty hard because the sample sizes are small. Go ahead. Well, yeah, so I don't have a big AIC or BIC table information criteria, but in every case, the Laplace distribution is the best fit. And when we do parameter estimation, on almost all cases, we get an exponent alpha very close to 1. A few cases it goes down to 0.8, 0.9. I mean, a couple cases it goes up to 1.2, but actually about 6 of those cases, It's just spot on 1, 1.03, 0.98, something like that. So very strongly Laplacian. So biodiversity, there's great variability, increases and decreases, but the variation is actually a Laplace. It's also known as a double exponential, 2 exponentials back to back, which implies 2 things. There's much more stasis, near zero change, and much more extreme change, fat-tailed change. I'm going to spare you the dive into probability theory, but there are a number of mechanisms you can get a Laplace distribution. If you have heterogeneity in your variance structure, for example, heterogeneity varies across space, or variance varies across space, or variance varies across species, that can lead to a Laplace instead of a normal distribution. If you have a Matthews effect where the rich get richer amidst a context of finite resources, this can lead to the Boltzmann distribution that Jeff alluded to, and ultimately to something that looks like a Laplace distribution. You can also have a regulation. You may know the Ornstein-Uhlenbeck is an extension of the random walk, where the regulation is proportional to how far you are from the center. If you have a constant regulation, you're always pushed back to the center, but it not dependent on how far you are from the center, you actually end up with a Laplace distribution. And I would argue this is not just, a mathematical artifact, but it's really important from a conservation point of view. We need to stop worrying about the global average, the total suite of species, and we really need to understand that most species are pretty close to zero, and there's a small group of species that are showing extreme changes, and that those are the ones that we need to focus on. Of course, classic species-based conservation has been doing that, but as we've been increasingly moving towards triage and global and community-based conservation, we've started to obsess about the mean trend. And I think the mean trend is actually not a very informative thing. So I think, yeah, that's my last slide. So I'll get to stop there. Yep. And love to have a conversation.
Great. Do it. Okay, well, I'm going to step back.
Yeah. Oh, that's it. So
okay,
people to ask questions.
If there are them. Okay, have at it. Who wants to ask Brian something? Ah, there you go. Go ahead, Milan.
Okay, can you hear me?
Yes,
we can.
Yeah. Okay, Ted, thank you very much for the interesting presentation. I would maybe first have a comment or question on the first part of the talk, in which you concluded that the local diversity is a kind of, let's say, stochastic reflection of the regional diversity on the larger scale. Well, my experience is that if there is a landscape that has high gamma diversity, then in this landscape you find communities that are rich and that are poor. So this reflection of regional diversity doesn't work everywhere. It only works in some areas. And I would rather say that local diversity reflects regional diversity only if we look at the richest communities within the region, not on the mean diversity of the whole region. So what do you think about it?
I think some of your observations are not inconsistent with this sample. I mean, the strong variation in richness, I guess my first question would be how much of that is explained by the abundance of the different communities, right? If you have high productivity and low productivity communities, you probably have more individuals in the high productivity than the low productivity, and that could still be a sample. To the extent it's not explained by abundance, at that point, yes, I think you do have to start to invoke some mechanisms beyond sampling. I think as far as your second point about, only rich local communities are reflecting the regional pool, I don't know. I guess I'd have to dive into that deeper with you, because I think, when you have a small sample from a large regional community, you're going to leave a lot of things out, and and you start to have asking questions about whether the same things always get left out and some other things like that. But could be hard to differentiate that from a sampling. But there are things that you could test to see if it is different from random sampling.
Okay.
Okay, great, thanks. Jirgi, you're next.
Yes, let me switch on my mic. Maybe you want to see me. Hi. I have a question regarding the very early introduction of the term biodiversity. I understand that we as biologists need to operationalize, so we need to get it down to something we can actually measure. But I would claim that originally the term biodiversity was meant to be multifaceted. It was meant to be kind of fuzzy. Otherwise, we are back, and actually we by looking at it through the species diversity, species richness, we are back to a term of species diversity which has been here since at the latest, well established in the 1960s, around even longer. So the question is, shouldn't we, when we talk about biodiversity, shouldn't we really consider that this is including, as also the political kind of definition of Rio says, it has its genetic component, it has ecosystem component or landscape component, and when one can also look at it from different angles. So let's say functional diversity, traits, which is quite fashionable at the moment. So it's not just about species and species richness, right?
No, I completely agree. I mean, like you said, it came out of a policy arena. I think it was a pretty inspired term in a policy arena. It seems to, bio and diversity, they're things that capture people's attention, and it's worked very well. And I think, I think about E.O. Wilson trying to put that seminar together, and I forget, I think he had about 20 speakers, right? I'm sure those 20 speakers all had different definitions of what they were thinking about and talking about. And so he needed an umbrella term to capture even the scientific point of view, let alone the policy point of view. So I'm definitely not arguing that we should oversimplify biodiversity back down to species richness. But on the other hand, we do have conversations with politicians and policymakers about what's happening to biodiversity. Is it increasing or decreasing? What happens if we, build these suburbs? Is biodiversity going to go up or down? So we do have some need to operationalize it too. And, I certainly have argued that if we operationalize it, it needs to be complex, right? I keep talking about alpha diversity, beta diversity, compositional change, abundance of biomass, and all of those things at different scales. Yeah, I completely agree. Biodiversity is intentionally vague, and it's a good thing that it's vague. I do think as scientists, if we want to have claims about what's, if we want to have a scientific claim about what's happening to biodiversity, we do have to step back and figure out how to operationalize it. But another piece that I sometimes give a talk on is how we communicate about biodiversity. And I think one of the implications of biodiversity being a soft term that exists in the policy arena is there are things that we can't quantify that are important about biodiversity too, right? I had it was simplistic, but I had the color figure of the warblers, and then I had the black and white figure, right? And, I believe all those warblers are native, but if one of those warblers was invasive and I told you that, you probably would care less about that species than the other species, right? And so I think biodiversity picks up some human values and some normative social values that's important. And as part of why it's effective. But I think as scientists, we need to start recognizing it's not just species richness or beta diversity, but there are some normative values behind the term biodiversity. And I think scientists, ecologists and social scientists need to spend some time talking to each other and trying to understand. There's an interesting conversation going on. Half of the ecologists immediately throw the invasive species out and don't count that towards species richness. And the other half do count it. And that's why regional richness is going up, is because of invasive species. And is that a good thing or a bad thing? And, now I'm using the word good and bad, which is a human value. And so I think we got to bring the whole complexity into it, but we also have to be able to measure things. So I'm trying to have my cake and eat it too. I'm not giving you a clear answer, but I agree with you.
Thank you. If I may, I have another question regarding the conclusion part, basically, of your talk about the trends. Let's say general trends not being really so much of relevance for conservation. I think that's what many hands-on conservationists feel, because they are concerned about their specific species for good reasons. But on the other hand, so what would you then reply to people who would say, yes, but we have this insect decline crisis and we have an overfishing? So, I mean, these are general problems which might have big ecological impacts. And of course, at the same time, they also boil down to losing species in the end.
Yep. So, right. So if you think about the figure I showed on the LPI, right, with the extreme changes and then the big group that's not changing, an interesting question is, can we describe that group that's showing big changes, right? Are they all in the tropics? Are they all fish? Are they all big body sized, right? Can we start to put some more general descriptions on that group? And I do think actually you put your finger on two groups that fish and insects that do show some pretty clear systematic trends. You know, I'm not aware so much in the species diversity of fish. We haven't seen a lot of, knock on wood, global extinctions yet, but clearly the biomass of fish, and it's very preferentially focused at the upper end of the food chain, have changed drastically, right? So I think we have to acknowledge that fisheries are a special case. I think trees, again, I don't think the extinction, I'm not aware of, you know, large numbers as a fraction of total trees of extinction, but again, we've taken 50% of the biomass of trees out. So that's something we have to recognize. And insects, I think there are a number of fairly consistent studies that do show a trend in decline in insects, and whether that's too many pesticides running around, I think maybe that's the answer for bees, which is a much narrower group, but whether there's too many pesticides running around, or, you know, you may know Mike Kaspari just published the dilution hypothesis, right? Increased CO2 makes food, makes plants into basically junk food, too much sugar and not enough carbon, and not enough nitrogen and phosphorus and the other nutrients that are needed. And so that could be driving insects down. So I think you're right. You certainly need to focus on those species showing big trends, but it's also an interesting question. Are there groups or places where we're seeing big trends? Mostly we haven't seen places with big trends, not marine versus terrestrial, tropics versus temperate, but there are some groups. I think insects and fish are two that stand out to me as well.
Great. Does anyone else have a question out there in the audience? Well, while we're waiting, I'm intrigued by Laplace, by the Laplace distributions and how often not only are they showing up, but with the same parameter value. And, you know, I'm interested in sort of universality of patterns, you know, beyond ecology. Have you looked at any non-human systems, other complex systems, to see what the characteristic Laplace values would be?
Great question. No, I haven't. Maybe we should write that paper, Jeff.
Okay. Well, and I go back to some early complex system stuff that shows that the actual, you know, the distribution of event sizes in ecological systems are actually different from many complex systems. They're more curvilinear. They're not power law. So there are some indications that biological systems are a unique type of complex system, sort of statistics and statistical mechanics. And I'm just really curious if Laplace, whether that's universal across or whether there's whether that's a biologically unique signal. So anyhow, okay.
So I did forget. I mean, the one place where Laplace has existed for a long time is in economics.
Oh, sure.
Right? So if you, whether that's a biological system or not, right, is a whole nother conversation.
Yeah.
But the econophysicists have been on top of the Laplace for 20 years. And so, again, the core claim is rates of change or trend lines are what show Laplace. And of course, if you look at stock market prices, if you look at GDP of countries, these things all show a Laplacian distribution of trend lines.
Okay. Well, interesting. And of course, there's a linkage between GDP to individual energy use that's universal. So there's some ways that these could be tied together. So,
Yeah.
Milan, did you want to ask another question? Yes, you did. Go.
Yes, if I can. I have a question regarding those studies that show no change in alpha diversity over time. Whether you are sure that there are no biases in the data, because I'm also working with data from repeated surveys in plant communities in my case. And what I'm seeing often that the new surveys or repeated surveys of previous surveys are often done with species lists from the previous surveys. So the researchers go to the field, they already have the list of species that should be done there. They find some species there, then they check in the list whether something from the previous survey is missing. So with this approach, there is a bias towards detecting higher species richness in the newer surveys, and we also see it in data from permanent plots. If permanent plot is sampled for the first time, then the group of researchers doesn't know all the plants very well, so they can overlook something, and over time they learn more and more. And now we see no net change in the data over time. But what if alpha diversity is actually decreasing, but the data are biased towards increasing species richness, and the result is the net change? Did you check your data whether there would be such an effect?
It's certainly possible. We spent a lot of time trying to think it through. These data sets are not list-based. They're community surveys. So, in one case, they're birds, right? So people typically go out and they count every bird seen or heard. In another case, they're plankton toes or they're bottom trawls or they're all kinds of different surveys. But they're counting every individual in a community and assigning them to a species, because one of our requirements is we actually had to have the abundance of each species through time. So they're counting every individual in identifying species. I don't think they would have. I've also done quite a bit of work with checklist data, and those do tend to have more biases. Taxonomy is not perfect, especially field taxonomy. Could people be misidentifying species? Yes. If they're misidentifying them, is there a bias towards a new? I don't know. I can't say absolutely no, but I think given the type of data where they're counting every individual in a community, the biases are probably not huge.
Okay, yeah, thank you. I didn't know that you have only this kind of data, because I'm a plant ecologist, and we rarely have individuals except for the studies on trees where they count individuals. Yeah. Okay.
Yep.
Thanks.
Well, and I'll just point out, you mentioned taxonomic error. I mean, one of the things that have been looking at is the fact is that a lot of traditional taxonomy is actually not based on empiricism. It's someone declaring themselves an expert and then saying this is a species. And when you go back to actually test those hypotheses, at least in the land snails that I work with, it's a 50% error rate. Oh my God. And it turns out those errors are scale dependent. Traditional taxonomists are more likely to oversplit at the local scale because they think they know everything, and they're more likely to overlump at large continental scales where they feel like they're not an expert anymore and they're just assuming there's climal variation. And it actually affects in a sort of biases our estimate of turnover. We actually are underestimating turnover rates, the churn through space, because of the fact that taxonomy is still stuck in the pre-scientific era. So there's a whole nother layer of error here, and it's an observational psychological issue. And as you move towards empirically based taxonomic decision making, it really does upend things because we were wrong a lot of the time. So something else, I don't know how that would affect any of these data, but I certainly know that there is a bias that comes in, and it's scale dependent.
Yeah, I mean, these were local, so I imagine they might have less of the error that you're talking about. But I will say, I mean, the main bias we have is just spatial, right?
Yeah.
We have a lot of data points in North America and Europe. We have
Yeah.
some in South America, very few in Africa, almost none in East Asia, Russia.
Sure.
So, but of course, every other data set has those biases, and we're not noticing big differences, right? We subset our data into the tropics, and it doesn't show different trend lines.
Right. Right. But like I said, it's something to be aware of, is that opinion-based taxonomy may be underestimating large-scale turnover.
Yep.
And overestimating the number of species present in a local pool. So, any other questions out there? What a quiet group. There's just been 3 of us having a discussion. Nobody else out there? Okay, well, Brian's a busy person. I don't want to have him sit around and just chew the fat. We've got other things to do. Shall we wrap this up then? Is there anything else out there? Okay, I'm calling it. Brian, thanks for being willing to come and present this really interesting work with us, and I'm sure we'll have lots of discussions in the department over the next week or two about just ideas that it inspired. So thanks much for sharing this with us. We appreciate it.
Yeah, thanks for the opportunity. Feel free to shoot me an email if somebody does come up with a question, and I hope I get there sooner rather than later.
Okay, I do too. Okay. Well, take care, everyone. Thanks for attending.
Thank you. Bye-bye.